Hi everyone, hello, how are you doing? I hope you're doing good. So, welcome to another video of Chapter 9 Human Health and Disease. In this video, we will be talking about what are the different ways in which an antibody interacts with the antigen and tries to get away, with the antigen. And then we will be learning about the structure of the antibody in detail. So now antigen and antibody can interact and the antibody can get away with the antigen in different ways, in different mechanism. The first one is precipitation, wherein if the pathogen which has entered the body is soluble in nature, the environment is created in such a way, the antibody creates an environment wherein the pathogen will become, it causes the pathogen to become insoluble, and then it will automatically precipitate out, and then that precipitate can be further phagocytosed by any phagocytic cells like macrophage or natural killer cells, etc. Another way in which an antibody can interact with the antigen and kill the antigen is by agglutination. What happens in agglutination? Here, all the cellular pathogens can be clumped together for removal of that particular of those pathogenic microorganisms. Okay, so they will be clustered and clumped together, and then those clots can be then further phagocytosed by any phagocytic cell like macrophages, natural killer cells. Another way in which an antigen can be, destroyed by the antibody is through neutralization. Antibodies can occlude the pathogenic regions by secreting exotoxins. Okay, so these antibodies can produce certain chemicals or certain molecules which will have an effect on the antigen in such a way that there is neutralization of the antigen. They are no longer effective in binding to other healthy cells. So the antigen will become weak or it will become inactivated. So now in this case, it will not be able to bind to any other healthy cell because the antibodies have neutralized the antigens. Another is inflammation, wherein antibodies will trigger an inflammatory response within the body. That is, it is killing the antigen in such a way that the other cells get signaled, they get triggered, and now they are aware of, they are prepared for any kind of infection that might come in the way. Okay, so that is inflammation. And another type of mechanism of the antibody antigen interaction is by complement activation. These are another set of immune response molecules. These are basically proteins which will go and create holes in the infected cells. Okay, they will go and create holes, infected cells with the help of these perforins, granzymes. Eventually, what will happen? The infected cells will undergo lysis. They will undergo destruction. So the cell also this is destroyed, and the pathogen inside the cell will also get eventually destroyed. So this is by complement activation. I will give you an image of all these, how they look like over here. So if you see over here, neutralization, that is the antibody is coating the virus or neutralizing the binding sites of the virus in such a way that it is not able to bind to any other healthy cells. Okay, so blocking the viral binding sites, or if it is a bacteria, then it will coat the bacteria completely so that the bacteria cannot go and bind to any healthy cells. Okay, thereby restricting the infection. Second one is agglutination, as we have just saw that the antigen and antibody will be clumped together. The bacteria will be clumped together and they will form clots. And then once the clot is formed, they will be destroyed by phagocytosis. Even here, next step is phagocytosis. Then precipitation, the antigen molecules will be made insoluble. Okay, and then it can be the antigen antibody complex can be made insoluble by the antibody and other immune cells, and then they will be removed out by phagocytosis, by precipitation. And finally, the complement system can get activated. These are some protein molecules which will create hole in the infected the cells, and then these infected cells will release the destroying molecules, and then finally the cell will undergo lysis. So these are the different ways in which an antigen antibody can interact and bring about the destruction of the antigen and the infected cell with the antigen. Okay. Moving to how the antibody looks like in detail, we cannot study the structure of antigen because antigen can be of various different structures. Okay, that is something which is foreign coming from outside. But definitely we have to know how a typical antibody looks like. So this is how a typical antibody looks like. You see a Y-shaped structure. This position is where the antigen can bind. Let's say this is the antigen, foreign substance, bacteria or virus, and here is the binding site. Okay, so you see two chains or four chains over here. Another name for antibody is immunoglobulin, Ig. It is also known as immunoglobulin. Okay, it has four polypeptide chains. So the basic chemical composition of an antibody is proteinic in nature, polypeptide. Proteinic in nature, they are Y-shaped. Okay, they have two identical heavy chains. So the bigger chains are known as the heavy chain. This is your heavy chain. Yeah, and the shorter ones are your light chain. They are, what is the chemical composition of these chains? Basically, these are polypeptide chains. Two heavy chains, two light chains. Heavy and light chains are connected to each other by the disulfide bonds. So both of them are connected by, obviously, what will be the bond existing between them. We have studied this in 11th standard when we were doing the tertiary and quaternary structure of proteins. That is, the tertiary and the quaternary structure of proteins can be held together by disulfide bonds. Okay, so they are connected to by each other by disulfide bond, and both chains contain two different regions. You can see the light blue regions and the dark blue regions. So this is variable region, and this is known as, the darker ones is known as constant region. Now the name itself is telling you constant means what? Be it any type of antibody. See, antibody can be of different types depending upon what is the antigen. Okay, which antigen they need to destroy. So this, any type of antibody you take, this region will remain same. This region will remain constant. And depending upon which type of antibody is produced to kill a particular type of antigen, this variable region will change. Okay, so hence it is known as variable. It can differ from one type of antibody to another type of antibody. Okay, so these are the constant regions and the variable regions. Yeah, so this is how a particular typical antibody structure will look like. And once it binds to the antigen, so they form an antigen antibody complex. Now the binding site uh, uh, on the bacteria. Okay, so this is the bacteria, and they have certain determinants. They are known as epitope. So the binding site on the bacteria will be known as epitope, and the binding site on the antibody will be known as paratope. So the paratope and the epitope will come together, and they will form an antigen antibody complex. And now once the antigen antibody complex is formed, so they can be removed or destroyed by any of these mechanisms, followed by either phagocytosis if these three is happening, and if complement system is activated, then the cell will automatically lyse. So that's all for this video. In the next video, we will be talking about the blood grouping and vaccination part. Thank you. Take care. See you.